Good evening, everybody. Dear colleagues and dear friends, I really do see some friends that have joined us here, and I thank you all for participating and coming on. This webinar is being recorded, so if you have any questions, you can go back over it, or at the end of my little talk here, I will unmute the microphones and you can ask me some questions, which maybe I will be able to answer. But at any rate, first let me say on behalf of my hardworking team and myself, I want to thank you all for the opportunity to share some of our work with you tonight. Now, in the interest of full disclosure, I would like to give you this brief note first, and I will pause for a minute so that everybody can read through it a little bit. As you can see, I'm a family physician and the medical director of the Paulina Medical Clinic since '83. The Paulina Medical Clinic is supporting the Kolisko Institute financially. I'm a co-founder and president of the Kolisko Institute. The work of the Institute is supported largely and principally, I might like to add, by tax-deductible donations and by the True Botanica Company. I am a co-founder and CEO of the True Botanica Company, and the company places all of its profits from the sale of its products in the service of the research and the educational activities of the Kolisko Institute. I intend tonight to explore what I would like to call a new law that was found in the behavior of serial high dilutions. I would also like to show some of the practical implications of the above principles, as for example, in the Kolisko validation. As a matter of logistics and housekeeping, I would also like to make the comment that for the rest of the webinar, I will use interchangeably the concept of high and ultra-high dilutions and homeopathic or anthroposophic potencies. So a substance that has been potentized up to the 10 to the minus 23 is called a high dilution. Anything that is above the Avogadro number, above 24, above 10 to the minus 24, is called ultra high. And that does not touch on the concept of low potencies and high potencies, of which there are many ways of looking at it and many controversies, I suppose. So let's start with this slide, where I would like to tell you that my interest in potentized remedies goes back to the time when I first met the work of Rudolf Steiner. For those of you who do not know him or his work, let me just mention the fact that his collected works encompass about 400 volumes. He founded the worldwide Waldorf school movement. He started biodynamic agriculture. He's the founder of the Anthroposophical Society, and he is best known for giving tremendous guidance in the development of spiritual life. He was a man of such wide-ranging capabilities that many of us who are following him have spent a lifetime trying to apply his insights into our practical life. Now, Rudolf Steiner had a great respect for homeopathic principles, especially when it came to the discovery, done essentially by Hahnemann, of potentized remedies, of the principle of dynamization, of serial dilution. And in the comprehensive medical course that I'm listing here, published currently by the Anthroposophic Press under the title of Introducing Anthroposophical Medicine. So in this first course that he gave for doctors, for physicians, for healers, about 20 lectures, he mentions there homeopathic principles in 7 of the 20 lectures. which is rather astonishing considering the wide-ranging topics that he is covering and his intent to get the doctors to have a better understanding of nature, of the human being, of the interactions between them. The slide also contains the dates of some other lectures that Rudolf Steiner gives, and I've noted those on the right lower side of the screen, where Rudolf Steiner talks about the work of Lily Kolisko, which we will go into in a moment. So listening to this and seeing the enormity of the grasp that the man had on life and on medical and pharmaceutical principles, it is not astonishing that his scientific coworker, a young woman, his student, called Lily Kolisko, would go to him to find the solution to a problem that she had in the treatment of a problem that was existing at the time. So in brief, in 1920 and 21, in southern Germany, there was a very significant outbreak of hoof and mouth disease in cattle. Rudolf Steiner was asked what his suggestion would be for a cure, and he proposed a highly specialized, a highly complicated remedy made out of coffee, which then would be potentized and then would be injected into the cattle, into the cows. Lily Kolisko then goes to him and essentially says, there are many potencies. Which one do I pick? So her question to Rudolf Steiner once again is, how do I find the best potency of your suggested remedy against the hoof and mouth disease? Now comes the history-making answer of Rudolf Steiner. Let seeds sprout in various dilutions of the remedy. You will then obtain a curve that will show you the vitalization process that occurs in the body of the cow. So we need to understand a couple of details here. Number one, he tells her specifically that the life force of the cow has enough similarity to the life force that is in the seeds, in the wheat seeds, that one can actually draw a conclusion from the seeds to the animal, from the plant to the animal. In general, this would be a concept that would be very difficult to accept for regular physicians, if you will, or scientists. But tonight, I think that I'm talking clearly to colleagues and friends that understand the concept of the vital force of the etheric body. In fact, Hahnemann makes that a cornerstone of his teachings also by simply saying that a homeopathic remedy will be healing in as much as it can correct an imbalance that occurs in the vital force. That's very crudely expressed, but it does give us a sense for the realm that we are in. Now, in anthroposophy, we call these life forces being part of the so-called etheric body. And at the level of life, whether it is the life in the plant, the animal, or the human being, there is a commonality that is the layer, the level of the etheric body, of the vital force. Now, through anthroposophy, of course, we understand that we have to keep separate from that the soul life, and we have to keep separate from that the manifestation in thinking and memory, etc., of the ego. But for right now, it suffices to say that we are looking at Rudolf Steiner pointing towards the life body and saying that will be an essential way to do what? That will be an essential way to notice whether a potency that you pick is actually active enough that it affects the life body, that it affects this life force. And the stronger it affects the life force, the more successful that remedy will be, in that case, against the hoof and mouth disease. So let's see what happened now. Lily Kolisko goes and she starts doing this work, and essentially she devoted the rest of her life. So from 1920 to 1976, that's 50 years of her life plus, to continuously expand and repeat this work and see where she's getting. And so what you would do is, just very simply and logically, is that you would take some containers and you would fill them with soil in which the seeds can grow. Now, whether that soil is some paper that you're wetting and allow the seeds to grow a little bit, or whether you just fill pots with earth and let plants grow, you make it possible for life forces, for biological material to expand, to grow. And then she would put into these various containers sequential potencies, so that you would start with potency 1, then 2, then 3, then 4, and so on. And you can go all the way until 50 or 200 or 500. Lily Kolisko does all of these kinds of experiments. And then you see what happens. One comment, incidentally, Rudolf Steiner, for reasons that are not important right now, looks at the decimal potency. So the one designated as X or D, which is why you will see that the rest of the work that we've done here will be expressed in that potency of decimals. Recently, we've started working also with the C potencies, with the centesimals, again, for other reasons that are not important right now. To a certain extent, I will come back to that at the end. If not, maybe somebody can ask me why we're going now into the C potencies. But to come back to the work that she's beginning, please note on the slide that there is a couple of possibilities of what you would normally expect. So if I have a toxic substance and I give it very concentrated, first potency, 1/10 of the substance, and then I dilute it and dilute it, I just keep on going with the potencies, what I would expect is that maybe in the beginning, the plants will not be able to grow. And then as I'm diluting and diluting, there would be a straight curve going towards more growth. So taller plants, heavier seeds, and so on. It's possible also that due to, who knows, another principle at work here, think of the homeopathic use of low potencies and high potencies, that as you're going into the higher potencies, the plants will suffer and their life body, their vital force will not like that higher potency. So we would expect a line that you can see here going downwards. It is also possible that the plants will thrive for a while, but then they will come under the influence of a potency that is not beneficial and they will not grow anymore. So these are the three basic possibilities that we would expect. In fact, what happens is something historically new that, as far as I know, had not ever been demonstrated before. And that is that when Lily Kolisko puts up, when she does her seed experiments, what she notices is that, to take here as an example of a long series that she did, look how under the influence of the 17th potency, the seeds are not developing. They're developing a little bit more under the influence of the 18th. The 19th and the 20th potency, they really develop wonderfully. They grow high, the rootlets are forming, the stalk is going up, only to again disappear on the 21st potency. The 22nd and 23rd are growing again very tall. 24th, nothing. 27, 28, almost no development at all. So this phenomenon she sees again and again. For example, when she uses crocus plants with potencies of iron sulfate, what you see here again is that, for example, water, which is the first pot control, is growing up to a certain point. The second potency, the D2, almost nothing at all. So you would think that that is a concentration that is killing the plant. 3, 4, 5, 6, 7, they're going very, very well, almost the same, only to come to the 15th potency and look at the 17th potency when again almost nothing happens, only for them to grow again. I think that everybody's beginning to see that what they've discovered here was that the sequence of potencies results in a wavy pattern, semi-sinusoidal pattern, whichever way you want to call it, but this is a rhythmical pattern that was not predicted before they actually did the experiment. I'm showing you one more slide here with gladiolas, where she used silver nitrate. Same principle. Look at the potency 9. The plant is almost shriveled. Potency 14, the plant is essentially dead, only under the influence of the next 3, 4 potencies to develop beautifully. At the 20th, again, it goes down. I think that the principle is clear. Lily Kolisko did hundreds of these experiments, and what she did always is then to make what Rudolf Steiner demanded, made graphs where she plotted the mean of the many plants that were growing under that experiment or the seed. Unfortunately, at the time, they were not using statistical analysis, so that has always been a criticism for her work. And later on, when we started to work in that field, that's the first thing that we ensured, that we would have a proper statistical validation. When we look at one of the curves, we can understand better what her more complete discoveries are. So you see here the hyacinth plant that grows under the influence again of silver nitrate. She makes an average of the means, she plots them on the graph, and now there's a couple of things that we need to learn from this. What she pays attention to is the fact that at the 12th potency and the 21st potency, right there where the red arrows are, she has a tremendous vitalizing effect. Now, in Rudolf Steiner's words, the graph shows that those 2 potencies would be the ones that would be most important to look at. Now, the other important thing is that when you see the first black arrow at D14, the plant has suppressed its growth almost completely. Now, there are 2 ways that one could look at the graph. One of them is to say that the first group of potencies between the control, if you will, and the D14 is one group that would probably correspond to what we call nowadays the low potencies. The next group corresponds from one black arrow to the next to 28, would correspond to the second zone, the second group, which we would call the high potencies. So the red arrows allow us to know which one is the highest effective one in the lower potencies, which one is the highest effective one in the high potencies. That's what she looked at. She looked at the most beneficial. That was her number 1 concern in all her graphs, more or less. She looked at zoning so that she could distinguish between the potencies, once again, that are low and the ones that are middle or high. We will get into that detail a little bit later. And she then determined that the lowest potencies would be like a barrier, like a changing place from one zone to the other. Now, I will go into that later, but one of the things that we found very significant and that we've been pondering a lot, and that is an opportunity for me here to say that all this research is very, very recent, even though we've been doing it for now probably about 10 years. But I just had to explain to a friend recently what our position is. And anything to do with anthroposophy and to do with these serial potencies and the work of Rudolf Steiner is the rough equivalent of Galvani finding out that if you put two chemicals together, electrical current will flow between them, and that led to the development and to the discovery of the battery. Well, from the Galvani of the 1700s, 1780 or so, to the electric car, there is a long, long way. And that way is not just 200 years or so, but rather it has also literally millions of scientists working and cooperating and developing. So we can only hope that this is the beginning and a lot more research will occur. So what have we added to this? I'm starting this already now, although I will talk about it later again. We have added the looking at the significance of those points that are at D7 and D16, so the green arrows, yeah? And those are roughly at the same level that the water alone is. Meaning what? That at those potencies, you could make the argument that that potency is so weak, that is so insignificant to the life force, that there is no change. So whether you have that potency or water, it has the same effect on the plant. Now, there's another way to look at it, of course, and that is that once you have that zero potency in the realm of the high potencies, that shows you, or even at the low potencies, that both of those green arrows show you a balance that that potency can do at that particular moment. So it's neither, let's put it this way, it's neither hyperactive nor hypoactive, but rather that it has a balance. So it's not necessarily, we're beginning to think that even those 0 points on the control line, that even those may not be insignificant. But I'm sure that you can understand our enthusiasm that you have the beginnings here of a totally new way of being to distinguish what role various potencies have. I just want to show you a couple of examples of the work that she's done. Beyond that, here she's looking at horse chestnuts, various potencies. Anthroposophical doctors especially consider a potency close to D30 to be very active. Look at the dip in the middle of the screen there, around, you see the 30 at the bottom. Look at the significant dip there. So that is not a potency that I would use in order to vitalize the system. Here we have a work that she did where she looked at potencies between 0 control and 60 on the right side, right? D60. And you will see there how the beginning of the high potencies is in the realm of 40. So 35 to 45, that's where you have a general peak in that rhythmical zone that we would call the high potencies. So that if you would want to give somebody a high coffee potency, you would want, and if you adhere to the view that potencies below, let's say 15, 20 are low potencies, and potencies up to about 30 or so are high potencies, then you suddenly see that in the normal course of treatment, you would actually need to go to a 40 and slightly above to actually have a very active coffee remedy for helping somebody to sleep, for example. This is another gladiola experiment that she does with tin chloride. And now I would like to show you a couple of the graphs that she did. Remember that I told you that she did hundreds of them, all of them collected in a book that I will show you in a minute. And I just want you to see how different the graphs of these various substances are. So what she's doing here is that she's using seeds, and she is watering them from D2 to D60 or so. And this is what the signature of that particular potentized substance looks like when it is being allowed to show what it does to the vital forces, to the etheric body. Totally different picture in Tinospora, another plant. This upwards moving general pattern, where indeed the high potencies have a much higher influence than the very low potencies. Now, the next one is berberis, where berberis in the lower potencies is much more active. The higher the potencies are, the more it suppresses the vital life. Question: is that an indication that indeed barberry, excuse me, that barberry is anti-inflammatory, that it actually suppresses the etheric forces, and that we can use that, and we do use it in our regular healing protocols? A different kind of comment that I would like to make is that some workers later on, already beginning with 1925, they had the question, well, what would happen if we would gather a lot of these curves together and we would get an overall average? So the question was, can we get some sort of a universal curve that would allow us to then not have to do every single substance, which clearly involves a lot of work, right? And what you can see here is something that is observed actually a lot of times, which is that at around the 15th decimal, there is a dip, and there is an enhancement of the etheric forces in the places that I've marked at 7, 12, and 23. However, this is not always happening, and I can show you this one here. What I have done is to gather 97 of substances that she calculated curves for, where there was a clear indication of a control point, where there was where there was evidence that there was a similarity in how she did the experiment. So they were all seeds, they all had the same potencies, they were going from D2 to D60 and so on. And putting them all together and introducing the statistical significance, you can see how basically the various substances cancel each other out. Just think, for example, that you would put together this curve and this curve. Obviously, the two of them would cancel each other out. You would get something close to zero. And Lily Kolisko, indeed, was not happy at all with the idea of gathering a universal curve. She really was hoping that the curves would be accurate enough and done well enough that they would really reveal a signature about the substance itself, and that this would help the doctors in their therapeutic process. She wanted very much to work with doctors, but due to politics and life, that work never quite materialized, and she was sad about it her whole life. And you can see here why these universal curves would not work. Because if you take just the 30th potency, the 30th decimal, from the 97 curves, you can see how some of those plants, essentially plants and minerals, so those substances, you can see how some of the substances have such an influence on the etheric body of the plant that under the influence of a D30, they are 80% better developed under the influence of that particular potency. On the other extreme, some of them can nearly go down to 80% inhibition compared to control, and everything else is in between. This is one of the most significant graphs that I'm looking at, and that I find enormous to be extracted from her work to actually show that, yes, one really needs to know the signature of a substance for a particular potency. And she published this in 1959 in a book called The Physiologic and Physical Proof of the Effectiveness of the Smallest Entities. Rudolf Steiner suggested the word small entities, which is a very interesting concept. I will leave it at that for right now, but it's important to know how Rudolf Steiner thought of these potencies. All right, I would like to go back now to just a summary, if you will, of the historical fact, and that is that up to a certain point, certainly up to her point, two effects pharmacologically, dose response curves, were known. One of them, the blue line, is the pharmaceutical concept, which is that as I am decreasing the amount of a substance, its effect on the biological system will be less and less and less until it will be zero. Then, in the 1890s or so, I believe, two gentlemen, two scientists, Arndt and Schultz, came to the conclusion that there is a second model, a model which nowadays is called hormetic model, hormesis. And that is that you can decrease the amount of substance and notice a decrease in its effect on the biological system until you come to zero. At some point, you come to zero. But if you now decrease the amount of that substance even more, in our example here, 1/10, 100th, 1,000th, then the effect begins to go into the opposite direction. So instead of looking at a toxic effect, now you're looking at a invigorating effect, if you will. And this concept that you can go to a 0 point and then increase an effect to a point, that is the second pharmaceutical model that was existing. And interestingly enough, the research into these hormetic principles has increased over the years, as you can see here, something like 2,400% over what was there even as much as 20 years ago. So clearly there's an interest there that people are beginning to notice that, yes, there is this principle in life where you go beyond the 0 point and then something happens that is very interesting. But due to Lily Kolisko and Rudolf Steiner, we know that there's actually a third principle, if you will. And the third principle is that you can have this movement of semi-sinusoidal curve. a zigzagging rhythmical curve. That is a lawfulness that is added in the behavior, in the influence, if you will, of serially diluted substances over biological systems. So that's an important point up to here. This work was continued and it was discovered by others. I will go a little bit faster over this due to the time. And especially I want to mention Jacques Benveniste, the French professor who did his famous studies that were first put in Nature and Nature journal and then disavowed. And if you notice there, he can see clearly that there is an effect that the dilution has all the way to the 60th potency, but he doesn't emphasize the fact that it is all rhythmical. Look at that semi-sinusoidal curve that you see right in front of you. By the way, shockingly enough, this is also Jacques Benveniste after they hounded him, and eventually actually he died of heart disease because of the treatment that he received at the hand of the so-called science. Finally, I would like to mention here the work of a young medical student in Switzerland, Theodore Carmine. I've talked to him, and he was in medical school when he did this research, showing the influence of potentization going all the way to 100 and obtaining a perfect sinusoidal curve. He was so happy and he was so excited to be able to clearly demonstrate in an absolutely faultless scientific model that working with cell cultures and so on and so forth, having some powerful statistical parameters that he looked at. He was so excited about this that I think he thought that he was going to get a professorship at the university next. And in fact, more or less, he was thanked and told to, go and have a good life. And he is right now a family physician in Switzerland. This brings me to the work that we're doing at the Kolisko Institute. Now that you've seen the principles, I can go a little faster through this. We work with seeds, and we place them in these petri dishes, water them, each group of dishes with a sequential potency. Sometimes we do experiments that have over 30,000 seeds that have to be individually looked at, cared for, and allowed to grow in a controlled environment where the temperature is the same, the light is the same, etc. And then you can see the difference, how the curves are beginning to emerge. So this is water, and then look at the difference. For example, I've just picked two slides here between D23 and D33. And then they have to be weighed individually. And at the end, this is very important to us that we collect the sprouted seeds by now, and we dispose of them with a lot of respect, because they have given their life to us, so to speak, so that we can do those experiments. So these are the graphs that we're getting. So here you have one where we did carbo potencies from D4 or so to 80, and you can see that we're obtaining the same results that Lily Kolisko did. So you have the peak, the peak potencies at 7 and 21 and 38. You have also very low inhibitory potencies, and you have these either unhelpful or mysterious potencies that are on the same line as the control. And we have repeated these experiments many times. Lapis lazuli, for example, we repeated it on 3 different occasions, months apart, and you have here an overall curve of that substance, and the numbers 7, 12, and 19 emerge there, which would be the potencies that one would want to use if one would want to have a strengthening effect on the vital forces, or an effect overall of those particular potencies. And you can also see that we keep on getting these groups that are clearly defined. You have here another curve of plumbum met. Look at the difference between the first 20 potencies or so, and then what happens in the higher potencies. So lead, of course, will be inhibitory all the way to the 19th or 20th potency and begin to reverse its effect and go up high and be invigorating, so to speak, be reinforcing at 29, 30. Just showing you another curve here, the same principle, just to give you examples of the kind of work that we do. This is an interesting one. Viscum album quercus is the mistletoe plant that is used in the treatment of cancer. And look how the 25th potency is the one where clearly it is most inhibitory on the vital force. And we're using that knowledge actually in our practical therapies. Just examples. This is a reverse of the plumbum, if you will. Low potencies of Helleborus. Very vitalizing. High potencies. Again, statistically significant. High potencies already begin to inhibit. I just wanted this as a look to the future, where we have collected blood from a young male and female volunteer, and we can see there how the potencies are expressing themselves in a particular way. This is important because we are using homeopathically potentized blood to treat our patients, infections, allergies, etc. And it's always nice to have a sense, do we want to give a very low potency, or do we want to go higher into the 28-30 range? I'm now moving slowly into practical uses of this method. One of them that I've just picked here, completely neutral. We took two types of coffee that are available commercially, and we picked simply for this graph, we've picked the 6th and the 30th potency, so low and high, and you can see there that the blue one is still, if you want to put it this way, acting like a coffee at 6x. It's still strong enough there. But once you go to the high potency, it's very invigorating, vitalizing. The other one, I would say, is much weaker in its overall subtle effect on the body. It doesn't rise so high with the high potency. It rises a little bit still in the same manner in the low potency. It just gives you a perspective of what one could all use this method for. So up to now, we have seen that one can use the method for picking quote unquote, the best potency. One can determine that to see whether it's high or low, depending on what zone it is in, to see if one can now begin to say, how do various substances compare to one another? Not substances so much, even one can start comparing manufacturing processes, times of the year, constellations, comparisons are possible. Okay, so what I would like to suggest also is this, that some of these insights and some of this research was then taken from the Kolisko Institute, from the laboratory, and it was made available to create hopefully better quality products by True Botanica. And incidentally, our and I will emphasize that again later, our methods are available to any other company or group that would want to look into it. But for right now, this is an additional step in quality control of the True Botanica potentized remedies. Here is one example that we took Cinchona 6X, and we wanted to see if we offer that to patients. Is the Cinchona 6X any different from the water control? And in fact, it is statistically very different. It is 15 to 20% more stringent and more affecting than the control. We have done the same thing with many other substances. Here, for example, we've taken Iotum 6X, C6 and C15, and we're comparing them. They're relatively close, but look how you have to go all the way out to the 15th potency in order to have a clear difference, to have a statistically significant difference. The difference between control and C6 is not that much. Now, one comment quickly at this point, why do you see now sometimes that we do our experiments in the C system, in the centesimal system, as opposed to decimal? This is trying to accommodate the homeopathic healers that are used to the centesimal system. And while we have not gone yet up to 200C, we at least are beginning to make a number of products that are in the 15C range. At least that is known to have a very good effect in general by the French school. So we're going back and forth between X and C at this point. Here you have Ferrum arsenicosum, lower potency, vitalizing, higher potency, you begin to have the opposite effect. I will let you look at it. Here's another type of ferrum, Ferrum muriaticum. And now watch the difference between Ferrum muriaticum and Natrium muriaticum. C6, no difference. But once you go to 15, there it begins to really almost explode in its effect. I'm showing you this combination of graphs because I just want you to notice how in both of the charts, 6C was not any different than the water control. We tried it again and again and again, and we could not obtain a statistically significant difference. What that means is that we decided finally that we are going to offer as a product from True Botanica the Chamomile 7X instead of the 6X, even though 6 is a favorite, because we want to be able to say that the quality control revealed that the quality was not as optimal as it should be. And with that, dear friends and colleagues, I would like to thank you for listening and for having been present. I hope that this stimulates maybe the one and the other to think about potencies in a new way. I would like to thank also the team that you see here, because without their work, not much would be possible. They really are dedicated and enthusiastic. And let me tell you that the principals do not get any salary at all. And I would like to ask that you consider a donation every once in a while to continue this work. Scientifically, it's important that we do it. Politically, I don't even have to start. As you all know, there is a movement afoot by the FDA to say, how can we determine the quality of homeopathic remedies? How can we determine the safety? And here would be one method. So instead of us merely maintaining that we're doing all the right things and the good things and nobody's got a problem, which is true, we have the opportunity here not only to really begin a process of verifying quality, but also to understand better what we are actually dealing with. So maybe before Christmas or in the fall, you can think of this little tin can that we're holding out to you. I thank you for your attention, and I would like to see if there are any questions, and I will unmute the participants, and you have to speak up, and if I can, I will answer. Well, if there are no questions, then once again, I thank you for your attention, and I will close the webinar for tonight. I wish everybody a fantastic therapeutic success in your endeavors in the work with potentized remedies. Thank you. Good night.